Hi! So, the discharge of waste from municipal sewers is one of the most important water quality issues worldwide. It is of particular significance to sources of drinking water. Municipal sewers contains human feces, and water contaminated with these effluents may contain pathogenic organisms and consequently may be hazardous to human health if used as drinking water or in food preparation. Bacteria in water are, in general, not present individually but as clumps or in association with particulate matter. Coliforms are bacteria that are always present in the digestive tracts of animals, including humans, and are found in their waste. It is also found in plants and in soil material. The term total coliforms refers to a large group of gram-negative, rat-shaped bacteria that share several characteristics. The group includes thermotolerant coliforms and bacteria of fecal origin, as well as bacteria that may be isolated from environmental sources. Arat na!
Multiple tube fermentation. The technique has been used for the analysis of drinking water for many years with satisfactory results. It is the only procedure that can be used if water samples are very turbid or if semi-solids such as sediments or sludge are to be analyzed. The procedure followed is fundamental to bacteriological analysis, and the test is used in many countries. It is customary to report the results of the multiple fermentation tube test for coliforms as a most probable number or MPN index. This is an index of the number of coliform bacteria that, more probably than any other number, would give the results shown by the test. It is not a count of the actual number of indicator bacteria present in the sample. As one of the three methods approved by the Environmental Protection Agency or EPA, the purpose of MTF is to ensure that drinking water is free from coliform formation. In this method, we'll look for the formation of gas in the test tubes. Coliform will produce a gas because they produce carbon dioxide when exposed to sugars such as lauryl tryptose. Multiple tube fermentation is performed in 3 steps. First, the presumptive test, second, the confirmed test, and third, the completed test. The following video demonstrates how to perform the presumptive test and MTF from a water sample taken at the canal. The procedure are as follows. First, prepare 15 test tubes, all with 9 ml of lauryl tryptose broth. Next, mix the water sample well. Then, shake it at least 25 times. Fourth, using aseptic technique, sterilize the test tube. With the help of a sterile pipette calibrated at 1 ml, grab the water sample and add it to the test tube. Repeat the process on all 5 test tubes from the first row. Remember to sterilize the test tubes as this will prevent foreign organisms from contaminating the test tubes. Then, label the first 5 test tubes with 10 raised to the power of 0 as these test tubes were not diluted. Although you can label the entire row, labeling each tube will prevent future confusion. For the second row, we need to prepare a dilution of 1 ml water sample into 9 ml of a buffer solution. Tap the tube or use a vortex to mix the water sample with the buffer. Label the dilution at 10 raised to the power of -1 to indicate that the substance has been diluted once. From the 10 raised to the power of -1 dilution, grab 1 ml and add it to the tubes in the second row which have 9 ml of LTB. Sterilize the test tubes before you add the sample and after you have added it. Add 1 ml of the dilution into 9 ml of L'Oreal tryptose broth. Repeat the process with all 5 tubes from the second row. Sterilize the dilution before setting it apart. Label the second row with 10 raised to the power of -1, indicating that the water sample added was diluted once. Label all 5 tubes or the entire second row. Grab the 10 raised to the power of -1 dilution and sterilize the tube. Sterilize the tube containing 9 ml of a buffer solution. Dilute 1 ml of the 10 raised to the power of -1 dilution into 9 ml of a buffer solution. Top the tube or use a vortex to mix it well. Label the tube as 10 raised to the power of -1 dilution, indicating that it has been diluted twice. Grab the 10 raised to the power of -2 dilution and sterilize the tube. From the third row, grab a tube with the 9 ml of L'Oreal tryptose broth and sterilize it. From the 10 raised to the power of -2 dilution, grab 1 ml of the sample and add it to the first tube of the third row. Repeat the process with the entire third row. Remember to sterilize the test tubes using aseptic technique. Then, label the third row with 10 raised to the power of -2. And label the tube with the following: date, lab number, water source, and method. Lastly, incubate the test tubes for 24 to 48 hours at about 32 degrees Celsius. Together with the supply of food and water, the incubator is the ideal growth environment for the coliform bacteria. In this environment, the bacteria will reproduce at a rate so as to double the population every 20 minutes. This means that a single coliform bacterium could give rise to a population of millions in less than a day. After 48 hours of incubation, the results of the presumptive test are recorded. The test tubes are inspected, looking for gas production. In drinking water samples, the actual production of bubbles should be rare in less than 5% of samples analyzed. If we see no gas bubble, The test is negative for total coliforms. At this point, our water is considered to be free of pathogens as well, and no further analysis is required. If bubbles are evident, we have a positive presumptive test. In that case, we are required to move to the second step in the multiple tube fermentation analysis, which is the confirmed test. A sample from each positive presumptive test tube is then transferred into a new test tube containing more sugar. Another ingredient, which is brilliant green bile. While several microorganisms may show up as positive under the conditions of presumptive test, only the total coliform bacteria will be able to produce carbon dioxide in the presence of the bile. That means the confirmed test is more selective for coliforms than the presumptive test. If no gas bubble is evident after an additional 48 hours of incubation, then the confirmed test is negative. This means that there are no total coliforms and thus no pathogens, and the analysis is concluded. If a bubble is apparent, then the test is positive for total coliforms. However, remember that fecal coliforms are of greater public health concern than the larger total coliform group because the fecal group normally is found in the intestines. This means that another test must be performed to determine if our total coliform positive sample also contains fecal coliforms. There are a number of different ways to make this determination, including the EC plus MUG technique. The EC stands for E. coli and the MUG stands for methyl and peripheral beta-D-glucuronide. It's a special ingredient that will fluoresce bright blue under an ultraviolet lamp if the most fecal coliform E. coli is present. In this event, the sample is not only positive for the total coliforms, but for the more critical fecal coliforms as well. This could lead to what is called an acute violation of the total coliform rule. The third step in the multiple tube fermentation analysis is the completed test. This is rarely conducted on drinking water samples because the presence or absence of total and or fecal coliform is all that is needed to determine the safety of our water supply. The completed test uses a number of advanced bacteriological techniques, including microscopic analysis to determine the actual identity of the microorganisms present.
Keidel method. Keidel method was developed by a brewer called Johann Keidel in 1883. The protocol is built on the principle that strong acid helps in the digestion of food so that it releases nitrogen, which can be determined by a suitable titration technique. The method consists essentially of transforming all nitrogen in a waste sample into ammonium sulfate by digestion with sulfuric acid, alkalizing this solution and determining the resulting ammonia by distilling it into a measured volume of standard acid, the excess of which is determined by titration. In the next part is the experiment of determining the nitrogen content of the soil using Keidel method. Chemical and reagents used: sodium hydroxide, boric acid, potassium permanganate, methyl red indicator, bromocresol green indicator, 0.01 normal sulfuric acid, 0.01 normal sodium carbonate, nitrogen distillation unit, 5% pure ethanol, and 20 grams of air-dried soil. In preparation of mix indicator, we need 0.2 grams of methyl red indicator to be dissolved in 100 milliliter of 5% pure ethanol, and keep it in a separated bottle. In the time of analysis, 5 ml of each indicator is mixed and used for nitrogen analysis. Procedure: first, add 20 grams of air-dried soil in a distillation tube. Then add 100 milliliter of potassium permanganate in the tube. take 20 ml of boric acid solution in a conical flask. Then add 2 to 3 drops of mix indicator in the boric acid solution. A pink color will appear in the solution. This receiver conical flask is connected to the outlet of a condenser. The tube must be dipped in the solution of boric acid and mix indicator in the conical flask. Then keep the distillation tube on stand on the nitrogen analyzer. Put the other tube of the condenser inside the distillation tube. Then close the door of the distillation unit. Next, run the device and press the key of alkaline and add 100 ml of sodium hydroxide of 2.5% potassium permanganate oxides. It reacts with water to form ammonium compound and then ammonia gas. The ammonia gas being liberated, this gas is absorbed by boric acid solution in the reservoir conical flask. About 100 ml of distillate is to be collected. The color of indicator is changed from pink to green. Then, remove the conical flask and switch off the distillation machine. A blank sample without soil should also run simultaneously without taking soil sample. The solution is then titrated with standard sulfuric acid solution. The green changes from green color to pink. Close the burette and read the volume or acid required for titration. Important precaution that should be taken care of. The delivery of tube of a condenser should be a dip in the solution of conical flask. Conical flask should be removed fast after the distillation process. All the joints of the distillation assembly should be airtight. Observation and results. The calculation of nitrogen is based on taking a simple equation. The net equation of 2 ammonium hydroxide with the addition of sulfuric acid will yield a product of ammonium sulfate with water as byproduct. 98 grams of H2SO4 or 1 liter of 2 normal H2SO4 is equal to 28 grams nitrogen, or 1 ml of 0.02 normal H2SO4 is equals to 0.00028 grams nitrogen. Thus, the available or mineralizable nitrogen in soil in kilogram per hectare is equals to S minus B multiplied by 0.00028 times 10 raised to the power of 6 multiplied by 2.24 over 20 times 10 raised to the power of 6, where S and B stands for the titer values of sample and blank respectively. Interpretation of results. Results related to available nitrogen in kilogram per hectare can be interpreted as 150 to 250 kg per hectare belongs to the lower level of available nitrogen. 250 to 400 kilograms per hectare belongs to the medium level of available nitrogen, while 400 to 600 kilograms per hectare belong to the high level of available nitrogen.
Pure culture technique. The term pure culture refers to a population or colony of cells growing in the absence of other species or types. The idea that is, if an agar plate is inoculated with only one species, and there is no contamination, then each colony on a plate will consist of genetically identical cells have come from a single progenitor or parent cell. Pure culture technique allows us to isolate one species from a mixed culture. It is a useful tool that helps us to obtain a single kind of organism from a mixed culture. There are two common methods of pure culture. Both of these methods result in individual colonies that are isolated from a mixed culture. The strict plate method and the pour plate method will be discussed after the discussion of the preparation of the agar plate. Preparation of agar plate. First, weigh the agar media and mix with distilled water. Next, sterilize the media according to the manufacturer's instruction. For sterilization, boil the agar at 100 degrees Celsius. Some need autoclaving at 121 degrees Celsius and 15 pounds for 15 minutes. Others need to be boiled in a microwave oven for 2 minutes. Then, pour the hot media in sterile petri dishes and let it cool to solidify the agar media. Incubate the newly prepared culture media for checking the contamination. Lastly, after the incubation, choose the fresh agar plates for testing.
Streak plate method. Streak plate method is a pure culture technique which helps in isolating desired colonies from contaminants. The procedure begins in sterilizing a wire loop in flame. Cool the sterilized loop by allowing it to stand for a few seconds. Dip the loop in the sample containing a mixture of bacteria. Depending on the sample, the loop may pick up hundreds or thousands of which are spread back and forth across the surface of the agar. Making uniform smear with the inoculum at the edge of the plate, sterilize the loop as mentioned above, and make streaks from the smear. Since there is thinning of inoculum during streaking, isolated colonies are formed at the ideal streak point where the spreading has separated the individual or group of cells at location where they can form individual colonies. The pattern of streaking varied with different type of streak plate technique. For example, quadrant streak, T-streak, and etc. During the incubation, the bacteria multiply and produce colonies. Intermittent flaming of the loop can be done during streaking to reduce the load of the organism so that there is a better chance of getting isolated colonies. Keep the plate covered as much as possible to avoid contamination. While streaking a section of the plate, try to keep the inoculating loop in contact with the surface of the agar at all times. Quadrant plate method. In order to obtain well-isolated discrete colonies, the quadrant streak technique should be used. This allows sequential dilution of the original microbial material or broth culture or colonies on a plate or slant over the entire surface of a fresh plate. In the following video is the demonstration of two ways on how to do a quadrant streak. One, using a wire loop, and the other is using a sterile stick. On sterile stick, on the back of the plate, take a marker and draw 3 lines. Then label the quadrants as 1, 2, 3, and 4. Remove the sterile stick from the tube. Turn the plates 90 degrees and spread the bacteria from quadrant 2 into quadrant 3. Note, be sure not to press too hard so you won't tear the agar. Discard the stick and get a fourth and final stick in the tube. Again, rotate the plate 90 degrees and take bacteria from quadrant 3 and spread them in quadrant 4. This time, spread them in the entire area without overlapping strokes. On sterile wire loop. We start with a sterile wire loop and pick a single colony in the agar plate. Open up the new plate and streak the colony into the quadrant 1. Re-sterilize the loop. Remember to let it cool for a few seconds before it touches the agar plate, otherwise you'll sizzle the bacteria. Now, without getting any new bacteria, you're going to streak from quadrant 1 to quadrant 2. Re-sterilize the loop and let it cool for a second. Repeat the procedure for quadrant 3. Streak from quadrant 2 to quadrant 3. Sterilize the loop, and then you're going to streak from quadrant 3 and spread all throughout of quadrant 4. Use up all the space that you have.
T-streak plate method. One type of streak plate is by preparing a 3-sector streak or T-streak. To do so, we begin by drawing the letter T on the agar plate and numbering them by 1, 2, and 3. You begin by taking your culture and spreading it in the first sector, going back and forth. Then from the first sector, streak into the second sector. When doing this, make sure that you do not cross back into the first sector of the plate. By doing this, that allows you to dilute or spread out your culture in the hopes of obtaining isolated colonies. From the second sector, we then streak into the third sector, making sure that we do not cross back into either the first sector or the second sector. The following video is the actual experiment on performing the T-streak plate technique in an agar plate.
Pour plate method. The pour plate technique can be used to determine the number of microbes in ml in a specimen. It has the advantage of not requiring previously prepared plates and is often used to assay bacterial contamination of foodstuffs. Pour plate method is one of the isolation methods used for counting the number of CFU present in a sample. What is CFU? Colony Forming Unit, or CFU, is a unit used to count the number of viable microorganisms. Now let's see how it is done. Materials: microbial culture diluted, petri dish, nutrient media, sterile pipette, incubator, colony counter. Procedure: prepare sample by serial dilution. Label the petri dish with the following: name of the culture, dilution fold, and date of inoculation. Note: include other details depending on your experiment. Inoculate 1 ml of sample using sterile pipette. Slowly pour the molten agar into the petri dish. Note: the temperature media should be 40 to 45 degrees Celsius. Gently rotate the plate. Allow the agar to solidify. Incubate at the required temperature. Then count the colony forming unit. Advantages of pour plate method. Useful for enumeration of viable organisms. Suitable for organisms present in low number. Aerobe and anaerobes can be enumerated. Disadvantages of pour plate method. Colonies will appear on surface and submerge in the agar. Colony morphology cannot be studied. The hot agar may injure cells, and it is time consuming. In coliform detection, detection of coliforms is used as an indicator of sanitary quality of water or as a general indicator of sanitary condition in the water and food processing environment. Coliform bacteria are important water quality indicators related to human health. Coliform bacteria are unlikely to cause illness but their presence in drinking water indicates the presence of pathogens in the water system. Now, why do we use coliforms instead of directly testing for pathogens? Total coliforms are used as a general indicator of potential contamination with pathogenic organisms. Because pathogenic bacteria in waste and polluted water are usually lower in numbers and are harder to isolate and identify than coliforms.
Nitrogen is one of the major elements required for life. It stimulates ground growth and produces the rich green color that is the characteristic of a healthy plant. Because of this, determination of nitrogen using Caldwell method in soil is essential for plant life. The rate of plant growth is proportional to the rate of nitrogen supply. If the soil is deficient in nitrogen, the plant becomes stunted and pale. However, we must always remember that an excess in nitrogen can damage the plants, just as over-fertilizing the lawn can damage and burn the grass. The isolation of bacteria in pure culture is important because it facilitates the application of recombinant DNA technology through the isolation of clones. In addition, microbial cultures are used to determine the type of organism, the abundance in the sample being tested, or both.
Wi-Fi, sci-fi.
How you broke my heart. Very good.
And are harder to isolate and identify than coliforms.